Hello, STEM students. This is Mr. Lima with one more remote class for science. And today is the Wednesday, March the 25th. We're talking about the cell cycle. We're continuing with the project that we started earlier this week with the mitosis and uh, cancer. I'm sorry, the mitosis and cancer project. We all worked on the activities for WISE website. And today I'm going to give you some definitions and um, some of the special structure and functions of the parts of the cell that are responsible for the cell division process or the cell cycle. Um, you know, so what we're actually doing is trying to understand as an essential question, how does sexual and asexual reproduction affect the passing of traits to offspring? So how the DNA can be divided and how it can bring the information to different cells um, throughout the entire cycle, especially during the division process. All right, and that's according to the North Carolina standards that you can see right there. Let's see if I can. Here we go. I can see full screen. So the idea of the class today is try to understand how actually we are how actually the cells can be divided and the cells will be able to organize and control all the cell division process. So important terms in karyotic cell division. Here what I bring to you are some vocabularies that I know it's important for you to know during this unit. And instead of taking notes, I just want you to pay attention because I have done the work for you already. So chromosome, when I talk about chromosome, I refer myself to thread-like structures that are composed of DNA plus protein. So every time we talk about chromosomes, the idea is to talk about thick pieces of DNA that are visually seen on microscopes, of course, but they are still seen during the process of cell duplication. They're made of, again, DNA plus protein. The second vocabulary that's important is the replication. A replication is a process whereby DNA is identically copied, of course, before cell division. It's important to remember the DNA is copied before cell starts to divide. It happens in a phase of the cell cycle we call interphase. And the end of this phase, but still interphase. We'll talk about more about this in a minute. So the cell division actually we call mitosis, which is when the nucleus starts dividing and all the processes that happen so the nucleus can divide. Okay. Cytokinesis, it's the division of the cytoplasm. After you duplicate your DNA, after your DNA has been copied, and the nucleus has divided inside the same cytoplasm, this cytoplasm needs to divide so we make two new cells, and that's what we call cytokinesis. And the last vocabulary here is a chromatin. Chromatin is a DNA in long strands that is thin and fibrous. It's invisible even under the microscope lens. It will condense into distinct chromosomes during the cell division. Well, but it's important to understand, why do we have to grow? You can grow too many cells. When we grow, we don't grow the cell. We make actually more cells. So one of the major characteristics of living things is the ability to grow. So what's the reason that our cells goes through the mitosis process? First of all, because it needs to grow. So we need more cells. An adult doesn't have bigger cells. They have more cells. And this picture can show you, if you overload the tissue with lots of cells, that's when cancer happens, uncontrolled division for the cell process. Okay? The next question that's important to understand is, when do cells divide? Well, they divide for reproduction, for replacement of damaged cells, for growth of new cells. And in replacement and growth, cell divisions, how should daughter cells compare to parent cell? Well, in this topic, I believe that it's important to highlight that when cells divide, they make identical cells. So that's why I go back to the idea of reproduction. That's why when you make new cells, we are still able to have the same tissues with the same organs, all of them working with the same function. In replacement damaged cells, because for different reasons we are constantly killing old cells and we have to replace them. And of course, individuals need to grow. All right. Well, in replacement of growth and cell divisions, how should daughter cells compare to the parent cell? They basically need to be identical. The daughter cells should be identical copies of the parent cell. There are three reasons why cells reproduce by asexual reproduction. We just talked about it: growth, repair, and replacement. This picture shows this skin cancer. It's a tumor, actually, which is basically an abnormal growth of the skin cell, most often develops on skin exposed to the sun. Every time you receive way more radiation that your cells can absorb, it affects the cell division process, and you start dividing way too fast in a way that you cannot control. Cell that reproduce by asexual reproduction reproduce what we call asexually. There are three main stages of the cell cycle. So the cell cycle, or the cell life cycle, is composed of basically three different stages. Stage number one is what we call the interphase. It's the longest stage. It's a preparation for cell division. Actually, yes, it is a preparation for cell division, but that's a very important phase because that's when the cells are working on their normal activities. So the cells of our pancreas, for example, is working on producing the pancreatic juice so the digestion can happen in the intestines. Your muscles are working so they can slide some proteins to contract and relax, so the functions of the muscle tissue can happen, providing movement. So interphase is the phase where cells are actively working on their specific functions. The second stage of the cell cycle is the mitosis. That's when the nucleus divides into two nuclei. Remember that you have one nucleus and the plural is nuclei. So one nucleus divided into two nuclei, each with the same number and kind of chromosomes as the parent cell. So the idea of the mitosis is the division of the nucleus. And we keep the same, it's a copy, so we keep the same number of chromosomes with the same information. And cytokinesis is the third and last stage of the cell cycle. That's when the cytoplasm divides, forming two distinct cells. Basically, you have two nuclei inside the cell, and that's the phase where you are, you are compressing. You can put it this way, the cell cytoplasm. So two new cells. Besides the fact they are identical, but they are new cells and they are formed that way. Okay. Well, so the interphase is quite long. That's where most of the cell's time are spent, and the cell division basically occurs very quickly. So here in this pizza graph, you can see that all this part here is interphase. All this part here is interphase. And you can see that the interphase is still divided into three different phases. We call G1, S, and G2. G1 is because of G for growth. So it's the first phase of growth. The cell actually increases in size. And then letter S is for synthesis, is a DNA synthesis. So that's when the DNA is copied. And then G2 is when the cell finishes growing. There's a second phase for growth. right? So interphase, the cell is working on their normal activities as much as growing and synthesizing DNA. During this short period of time here, that's when the mitosis happens. That's when the cell divides the nucleus, right? And then we're going to focus a little bit more on what happens inside this phase. 90% of the cell cycle is spent in this phase, in interphase. So we talk about interphase here in the slide. So 90% of the time of the cells, you find it during the interphase. G1, as I said, is the first growth phase. The S is the phase where DNA is being synthesized. That's when the DNA replication happens. And the G2 is the second phase for growth. All right. Important question. Why is it important that the nucleus divides in mitosis? Well, the nucleus stores the DNA, which is the information of life. Also, all the new cells need this information. So if you compare the DNA, the DNA could be as a chromatin. It can be seen as a chromatin during the interphase, where you cannot see it. A long strand of DNA. But we can also combine these and condense this information of DNA into shorter, compact pieces of DNA plus proteins we call chromosomes. So these are the chromosomes in humans. We have 46 chromosomes, and a chromosome is made of one chromatid and another chromatid. So there are two chromatids linked by this point we call centromere. The centromere right here. Okay. So chromosomes are defined as the genetic information that's passed on from one generation to the next through chromosomes. Every organism has a specific number of chromosomes. So humans has 46, carrots has 18, elephants has more than 300. So different organisms, they have different numbers of chromosomes. That's why it actually identifies different species. Chromosomes are not visible in most cells except in cell division, as I told you before. And why not? DNA and protein molecules that make up chromosomes is spread during the nucleus. That's why you cannot see it. It's spread. It's a micromolecule, so it's not possible to see it. At the beginning of the cell division, the chromosomes condense into compact visible structures. So here in this picture you can see the entire chromosome made of one chromatid, and the left side another chromatid, and each colorful band right here is a different gene that will regulate different parts of your physical or genetic information. For example, this could be the gene that tells you what's the color of your hair. This could be the gene that tells you what's the color of your eyes. This will be the gene that tells you how your tongue will be shaped like. So there are many different genes with different functions in parts of your DNA or referring to chromosomes. Before cell division or the interphase, each chromosome is replicated. I told you before, the chromosomes are replicated during the interphase. Each chromosome is made up of two identical sister chromatids held together by the centromere. So one chromatid right here, another chromatid right here, and this place in the middle, the black spot in the middle, that's what we call the centromere that holds the two chromatids to make the chromosome. Here's another picture where you can see, now it's in this picture you can see a real chromosome. So this is one chromatid, this is another chromatid, and the centromere in the middle. Right? These are human chromosomes. In this picture you can see the molecule of the DNA, and the DNA is a molecule that is made of different bases. That's the chemical composition of the DNA. When they combine together, they will be compact together and make the chromosomes. All right, and all this information is stored into one chromosome inside your nucleus, the nucleus of a cell. That's how it is structured. All right, so let's talk about each of the stages of the cell cycle. There are 3 stages, remember? Stage number 1, interphase. Stage number 2, mitosis. Stage number 3, cytokinesis. So let's talk about the interphase, which is stage number 1. I remember when we talk about the cells, structures, and functions, centrioles was one of the organelles that we mentioned that is participating into the cell reproduction or cell duplication. The centrioles are important because they will actually separate the different chromosomes in the cells according to the according to the information that they have. We're going to talk about that in a second. So the interphase is the longest part of the cell life. The cell carries out normal activities, as we talked about it before, and DNA is long in long strands called chromatin. So remember, chromosomes is when they are compacted. Chromatin is when they are long strands free inside the nucleus. And that's when the centrioles are made. The centrioles are produced in the end of the interphase. All right. So your nucleus is still here. Your DNA inside is duplicated, as you can see, red duplicating red, blue duplicating blue. That all this process happens during the interphase. And the centrioles, I'm sorry, the centrioles, they always work in pairs. Always work in pairs. Well, what happens in the late interphase? The late interphase, you still have your cell membrane and the cytoplasm. Your centrioles are duplicated, and the nucleus starts disappearing. Actually, the nucleus disappearing is the first sign that you are going to the cell duplication, or if you prefer, the mitosis. So when you start observing the nucleus disappearing inside the cell, that's a sign that the cell is not anymore in the interphase, but it's also entering in the prophase, which is the first phase of the mitosis. So chromatin doubles to form chromosomes. So the long strands of DNA now is getting compacted. Other cell parts also double. So the centrioles are doubling, everything is getting doubled. And editing of DNA to repair errors. During the interphase, we need to make sure that the DNA is being copied correctly. Otherwise, you're copying something wrong and you pass the wrong information, and maybe the individual will not be able to survive. So there is a kind of a check for the activity to see if the DNA has been copied correctly, and if it needs some repairs chemically, your organism is able to do it. Well, the interphase happens pretty much the same in animal and plant cells. Just remember, the plant cells, we do have the cell wall that's protecting the cell, and the cell membrane that's selecting the material that comes in and out. And animal cells, you only have the cell membrane. All right, and all this division process happens inside the nucleus. Remember. Well, before the cell starts dividing, remember the cell is coming out of the interphase. So the interphase is out. The cell stops working on the normal activities and start dividing, right? And that's when the first phase of division happens. Oh, here I have a slide that kind of summarizes all the stages of the cell cycle. So mitosis is only one continuous event, but it can be described as happening 4 different phases. Just another thing that's very important: mitosis is part of the cell cycle. So interphase that happens before mitosis is not part of mitosis. Okay. So again, the 4 phases that can be described during the mitosis are prophase. Pro means before, so what happens before actually division. Metaphase, meta means middle, and understand this in a second why this name. Anaphase, ana means movement, so it's the movement where there's a movement. I'm sorry, it's the phase where there's a movement of DNA inside a cell. And telophase, that's when you have the two cells being made. All right. In this slide, you can check by yourself. There is a link for animated cell cycle, and you can check how it happens. All the phase. So let's start with the prophase. Prophase is when your chromatids, right? You have remember that you have your chromosomes here in blue, another chromosome in blue. It has been duplicated before the prophase starts, as much as the other chromosome has been duplicated right here. But the left side is one chromatid or a sister chromatid, and the other side is another sister chromatid, right? The same happens here to the blue part of it, your DNA. Since it has been already copied, the idea is to bring one red and one blue to one edge of the cell, and one red and one blue to the other edge of the cell. So you can create a nucleus around this chromosome, then a nucleus around the other pairs of chromosomes here, and then you can just squeeze the cytoplasm, making two different cells. That's basically how it happens. Well, prophase is when the nucleus disappears. Important information for prophase is when the nucleus disappears. And then your centrioles can migrate to the poles of your cell and start pulling these DNAs or these chromosomes apart. So chromosomes coil and become visible. That's something that happens in prophase. Another thing is that centrioles or poles appear and begin to move to opposite end of the cell. The spindle fibers are these microtubule structures that will be connected from the centriole to the chromosomes, and then it drags to the edge of the cells. Nuclear membrane disappears. That's what I told you. That's the first thing that happens in a prophase, is the nuclear membrane disappearing. As part of the prophase as well, what happens in the cytoplasm? Your spindle fibers begin to form. So once you have, here you have the nucleus disappearing, the centrioles migrating to the poles, and then your spindles beginning to form. Right? The idea is to connect to the chromosomes and drag them to the edge of the cell. Spindle consists of microtubules arranged between the centrioles. Centrioles move apart due to the lengthening of these microtubules. So it grows and shortens, and then it can bring the chromosomes to the edge of the cell. Okay, how does prophase happen in animal and plant cells? Here is a prophase in an animal cell. Then you still can see part of the membrane that composes the nucleus. But what's important here, it's disappearing, of course. But what's important here is that the spindle fibers are being produced. So you can see chromosomes, a bunch of chromosomes here, and the spindle fibers being produced. In the plant cells, you see, in this image is very easy to just check the centrioles in one edge, and you probably have another centriole in the other edge, which can't be seen in this picture. The second phase is the metaphase. Well, just like I told you that prophase, pro means before, because that's the phase that comes before division. Metaphase, meta means middle, and during the metaphase, you will see now the centrioles in the edge of the cell, no nucleus anymore. Your spindle fibers are already connected, one side to the centriole, the other side to the chromosomes, to the centromere. So remember the centromere, that is combining the two chromatid sisters. that's the place where the spindle fiber connects as well. So that's an important chemical interaction part of your chromosomes. So the idea is to have all your chromosomes in the middle, and when the chromosomes are aligned in the middle, that's when you have the metaphase happening. That's when you identify it. So chromosomes line up in the middle of the cell on the spindle fibers, and that's pretty much it. So a picture in animal cells, you can see central on one side, central on the other side, spindle fibers connecting to the different types of chromosomes aligned in the middle of the cell. The same thing happens in a plant cell. Okay? And you can see in the side, on the cell here on the side is a different stage. Not all the cells are in the same stage at the same time. So this cell is in a metaphase, for example. This cell, it has the nucleus, and you cannot see the DNA. So this is the interphase. The cell is doing photosynthesis, for example, and doing its work. This cell here, though, already have part of the nucleus being reconstituted in both sides, and you have another division in the middle, which means that cytokinesis has happened. So this is the end of the cell cycle, making two different cells. So different cells, they are in different stages of their cell cycle. Phase number 3 is the anaphase. And during the anaphase, remember the chromosomes, they were aligned in the middle? Well, now during the anaphase, the same centriole with the spindle fiber is pulling one chromatid sister to one side, the other one is pulling the other chromatid sister to the other side, right? Remember, they are copies of each other. That's what has happened during the interphase. So now the cell that acquires this information on the DNA has actually the same information as this one because it's a copy of each other. All right? Chromatids, or halves of chromosomes, separate and begin to move to opposite ends of the cell. So ana means movement, and that's why anaphase is when you see the chromatids being moved. So this is a picture in animal cell when you already have, again, the centrioles, the spindle fibers in red, and now this part being dragged to the edge of the cell, and the same happens in the other side. In a plant cell, you see anaphase happening pretty much the same way. The last phase is the telophase. Well, telophase is the last phase of mitosis. Telophase happens when you basically already have dragged all the chromatid sisters. Remember, you have one chromatid, which is a sister of this one, for example, the blue and blue. This red is a sister of this one. So, and this red a sister of this one, and this blue a sister of this one. So they are all making two different nucleus with the same DNA information. The telophase happens when your nucleus starts appearing again, and your DNA is not compacted. So you're not talking about chromosomes anymore. We're talking about the chromatin being formed, right? A long band of DNA again. So the telophase is when the cell goes back to normal. If you want to put it this way. So chromatids finish separating, right? So this chromatid to one side and sister to this one, they are finally separated. They finish moving. The nuclear membrane reforms, and the chromatids unwind and become chromatin. All right, a long band of DNA. That's what characterizes the telophase. Pictures. So here you can see, of course, when you have the chromatids one side, the chromatids the other side, the centrioles are not here any longer. The spindle fibers, they also disappears. And this picture still are able to see the spindle fibers, but they will break apart. And you can also see a little squeeze here in the middle because that's when cytokinesis starts happening. So cytokinesis happens before telophase ends. And plant cell, you can see telophase happening. So the new nucleus in one side and new nucleus in the other side. Look at this picture. The cytokinesis is also happening here, but instead of squeezing the cell, it's impossible to squeeze the cell because of the cell wall, which is a very rigid structure. Well, Mother Nature is perfect, so it made the plants dividing, producing new cell wall from the middle, and growing until we reach the next cell wall right here, and then you can separate one cell from another. Right? It's different from animal cell that doesn't have a cell wall, so you can simply squeeze and separate the two cells. So I already talked about the interphase, stage 1, stage 2 mitosis with the 4 different phases: prophase, metaphase, anaphase, and telophase. And now cytokinesis is the last stage of the cell division. Cytoplasm and cell organelles divide evenly, right? That's when you see, as I told you, this cytoplasm being squeezed. All the different cell organelles there also being duplicated, and they divide evenly. 2 new cells are formed. So in the end, you can see the interphase as the phase where the cell is making its work. So we're talking about chromatin, where the DNA is very long, and you have the nucleus very visible. And then prophase, the nucleus starts disappearing, the chromatins kind of transform into chromosomes, and you start seeing the centrioles migrating to the edges of the cell. The metaphase, when the chromosomes are aligned in the middle. Here are the 2 centrioles that are connected to the centromere of the chromosomes. Then anaphase, when it drags the chromatid sister to one side and the chromatid sister on the other side, so we can make 2 different cells in the future. Telophase is when they stop moving and the squeezing starts happening, so you can make the 2 different cells. And cytokinesis basically is when this process happens, the squeezing process happens, the nucleus goes back to normal, the chromosomes are transformed into chromatins again, and each different cell starts a new interphase. Okay, that's how the cell cycle happens. The same thing happens into plants. However, remember that for telophase or the cytokinesis, basically, you're not able to squeeze the cell in the middle, so the division process starts from the middle and grows until we reach the cell wall. And then you have 2 different cells here made in the end. All right. This is a graph so you can understand the phases of the cell cycle. And my suggestion, actually my recommendation, is to watch the 2 videos that I made on Google Classroom. Please watch these videos. They have way more technology than I have, so it's really well explained. And do your quizzes for mitosis right after that. Thank you so much for your attention. I see you next time.